Hello and welcome to this lab overview for the Human Physiology Biology 206 Negative Feedback, Homeostasis, and Experimental Design Lab. So as always, a list of objectives that are available here as well as on the lab handout. I encourage you to read those to get an idea of what it is we're going to be focusing in on as the kind of outcomes associated with the lab this week. And keep in mind that this is consistent with the types of questions that you're going to be seeing on the lab exam. In addition to the questions at the end of the lab report, as well as the lab study guide. So just a quick reminder, I know we've already covered homeostasis within the lecture portion of the lab, but to make sure everybody's on the same page, homeostasis is kind of the body's response to maintaining this kind of steady state, to this steady internal environment within a physiological range. And recognize that when we're talking about homeostasis, it's not necessarily locked in place. It's not going to be, fixed and rigid. It's going to be a dynamic process, and so you're going to oscillate from being a little bit above to a little bit below to a little bit above, a little bit below, kind of oscillating around like that. And so what you're going to be looking at is some type of change that's going to be detected in response to some type of either internal or external stimulus. That change, that kind of modification within the internal environment or adjustment from kind of that internal set point is going to be perceived at some point by a receptor, and the type of receptor is going to vary depending upon what region of the body we're looking at. That receptor then is going to send information into a control center. The control center is going to make a decision ultimately as to whether or not this is a large enough departure from that homeostatic set point. And if it is, it's going to send a message out to an efferent or an effector organ, an effector structure that is going to respond to address that change. And generally what's going to happen is if something is too high, that change, the effector is going to push it back down to its normal state. If it's too low, that effector is going to change it, so bring it back up to where it should be. Okay, so the best example of that, the easiest one to visualize, is the thermostat. I think this has been covered at least in my lecture, I think in Dr. Wright's lecture as well. You've got a sensor within the thermostat here. You're going to have a set temperature point, and if the furnace is off and now it starts to cool, the thermostat is going to recognize that. It is going to send a message to the furnace to turn it on. So the furnace in this case is going to be the effector. I'm doing this backwards, I think. Okay, so one of the easiest examples to look at in terms of homeostasis and negative feedback is taking a look at the heating system within our house. And so we're going to see the situation over here where we're going to have a thermostat that's going to be our sensor. We're going to set it at a specific desired temperature. So what's going to happen is that as the house starts to cool, or the room starts to cool, that is going to drop, the room temperature is going to drop below the set point. So as the house is cooling, what's going to happen is the sensor within the thermostat is going to recognize that. It's going to send a signal to the furnace, and the furnace is going to turn on. And if the stimulus was a decrease in the room temperature, the response is the effector, our furnace over here, is going to turn on and warm the house. And so as that house warms, we're going to push that lower temperature back towards that set point. Once we reach that set point, that signal is going to stop going to our furnace, and we're basically going to see that the furnace is turned off on the opposite end. And so basically negative feedback, because the stimulus was a drop in the room temperature, the effector is raising the room temperature, and so we're minimizing that stimulus by addressing it in this way and maintaining homeostasis. Now within the body, a whole variety of different physiological measures, physiological parameters that we're going to be looking at. Same idea. We've got like the little teeter-totter over here. If the heart rate is going to be increasing, the receptor is going to recognize it, a control center is going to send it out. And then you're basically going to have an effect of slowing down the heart rate and returning it to homeostasis. And so when we take a look at this, regardless of the type of physiological system we're looking at, regardless of the parameters we're looking at. Most of the systems in terms of homeostasis are going to be using a negative feedback system like this. Now in lab this week, we're going to be looking at heart rate. And so the heart rate is going to be under antagonistic control. So antagonistic, we're going to have two components that are controlling the heart rate, and they're going to be antagonistic to one another. They're going to act opposite to one another. And so what we're going to see, and we'll talk about the heart later on within the semester, the heart is autorhythmic. It's going to beat on its own. And so if you watch the YouTube videos about heart transplants, you take the heart out of the chest, and it's going to be continually beating on its own, separated from the body for a period of time. And that's why we can do heart organ transplants. Now, the speed at which the heart is going to be contracting is going to be under the control of the autonomic nervous system, and we'll talk about that when we get to the nervous system. But within the autonomic nervous system, we're going to have sympathetic neurons associated with the fight or flight response. They're going to increase the heart rate. And then we're going to have parasympathetic neurons, so parasympathetic as opposed to sympathetic. The parasympathetic is your kind of rest and digest system, and that's going to have an effect of attempting to slow down the heart. And so what we're looking at with our heart rate is that balance that's going to be occurring between that antagonistic control between the sympathetic neurons increasing the heart rate and the parasympathetic neurons decreasing the heart rate. So we're going to think about this from the status of running an experiment. And so we need to come up with a hypothesis and build that into the lab activities we've got now. So the best way to come up with a hypothesis is to come up with a clear statement in the form of an if-then statement. And so we're going to be looking at heart rate in the laboratory this week in response to very short periods of exercise or very short levels of physical exertion. And so we can create an if-then statement. If a person does something, if a person exercises, then our hypothesis is if the person is exercising, then their heart rate will increase during that exercise period. Keep in mind that a hypothesis needs to be testable, and so we can test it and say, okay, our observations support the test, support our hypothesis, or reject our hypothesis. And it's going to be valuable regardless of whether we support or reject our hypothesis, because that is going to provide us with more information. So keep in mind that we're going to have two types of hypotheses, and again, this would have been covered in like a research methods class or Bio 242 reading or writing in biology. And so basically the two hypotheses is that there's going to be a research or alternate hypothesis, and that's there is a difference between groups. So the drug has an effect, there's a difference between the groups, so there's something that is going to be different, something that's going to be occurring as a result of the experiment that we're doing. So in this experiment, people exercising will have a higher heart rate than people recovering. And so, we take a look at it. We hook up a test subject right after they've exercised, measure their heart rate, monitor them for a period of time, and see if their heart rate immediately after exercise is higher than what it is after they've been recovering for a while. Now, in order to be able to do the statistics, we need to be able to see whether or not our observations made in the laboratory are going to be consistent with our hypothesis. And because we don't know what level of difference is going to be present, is the higher heart rate going to be 10% higher, 20% higher, double? We're not sure. And so mathematically, we are really going to be testing the null hypothesis. The null hypothesis says that there is no difference between groups. So there's no difference of the drug in this treatment, or there's no difference in this experiment between people, the heart rate of people exercising versus the person in a recovery or resting state. And so because of that, if there's no difference, then the heart rate at rest is going to be the same at heart rate immediately after or during exercise. And we need to be able to do that, because then we can say if there's no difference, we can take a look at our observations and say, is there a difference between our observations? Is there a difference that's going to be present? And if there's a difference, that is going to tell us that our null hypothesis is not correct. And so it's going to be our null hypothesis that we're testing with inferential statistics in this lab in the form of a t-test. Okay, so keep in mind that when we're running the experiments, we're going to be working with a dependent variable. A dependent variable is what the researcher is going to be measuring, counting, or observing. And it's what we think is going to be changed. So in this example, we've got a guy hitting a hammer with a carnival game, looking at how high the shuttlecock is shooting up. So in this case, the dependent variable is how high the shuttlecock is going up after the little lever has been pressed. Now in this case, we've got the same basic carnival game, but we've changed the person hitting it from the man to the woman. And so what we did there is we changed the independent variable. The independent variable is going to be the variable that the researcher is changing. And so if we're doing a drug study, we may have some individuals that are not exposed to the drug, other individuals that are exposed to the drug, and see if the drug has an effect. In this case, we're going to say, is there a difference between the man hitting the hammer onto this carnal device versus the woman? In our experiment in the lab, is exercise going to have an effect on the heart rate that is going to be different than an individual at rest? Okay, and so heart rate is going to be the dependent variable. The exercise versus rest is going to be the independent variable. Now keep in mind that when we run these experiments, we want to try to standardize everything as much as possible so that the only thing that we're testing is the difference between our independent variable. In this case, Homer Simpson is sitting down on the equivalent of our carnal device, but it's a different device. He's not using a hammer. All kinds of different things are going on. And so if we start introducing some of these other variables being present, it eliminates or makes it more difficult to determine whether or not the changes in our independent variable in this experiment, exercise versus rest, has the effect, or if it's some other thing that is going to be going on. Now, if we take a look at this, and again, just a quick overview, we've got a couple ways in which we can look at our data. The first is in the form of descriptive statistics. Descriptive statistics, just like the definition says, are going to be, in essence, numbers which are going to describe or kind of summarize the observations that we made. And so we may have an observation and we take 5 different measurements. And then from that, we are going to take a look at what is the mean or the average measurement that's going to be possible. What's going to be kind of the average response that we're observing. Now, ultimately, in a scientific report, we'll also look at the kind of distribution or dispersal of our observations. Not really going to focus in on that within human physiology, but keep in mind that you can often represent kind of the spread of the data, the distribution of the data in a variable called the standard deviation. And so we can give kind of an idea about how close or how far apart the different observations are from one another. Now, that's descriptive statistics, kind of summarize the observations that we're making. Inferential statistics is when we go and say, okay, we are going to do an experiment on the students in human physiology lab, looking at their heart rate after exercise and at rest, and we're going to try to say, okay, based on our observations of these students within the lab, can we then make statements about the general population? So we're not going to go through and have everybody in the world go through and exercise, take their heart rate, look at them at rest, take their heart rate. We're not even going to do that with everybody on campus. And so what we're going to do with inferential statistics is make an effort to take our observations from a small group, our sample over here, and extrapolate that into the broader population as a whole. Okay, so that's kind of the background introduction. Now we're going to take a look at what's actually going to be occurring within the lab this week. So again, hopefully download and complete your lab report. And so download the Google document. It's listed on your Canvas site. So it should say something like template, lab report number 1, make a copy and edit on your computer, submit to instructor when finished. Download that either onto your computer or make a copy onto your Google Drive. Name it. Not all of us are going to grade it online, and so if the instructor is going to download it onto their computer, they're going to have a lot of reports that say lab report number 1, and unless you put your name on it, they're not going to know exactly who that report is from. So if you name it, then we've got an idea who it is that submitted it if we download those. Okay, so the setup, and I'm preparing a YouTube video, so showing you the setup for our negative feedback lab. We're going to use the Power Lab system over here, hooked up to a pulse transducer, which is on the figure there. Follow the instructions in the ADI Introduction to Lab Chart 8 Student Protocol, and we're going to do the negative feedback lab. And we're basically, the hypothesis is we're going to say, is the heart rate different during recovery from physical activity versus at rest? And so we want to identify the independent variable, the dependent variable, and get a feeling for how those variables are going to interact with one another. And so the idea is exercise or at rest is going to affect the heart rate, and that heart rate then is going to be observable as we measure the heart rate in essence. So the independent variable ultimately is going to have an effect or not have an effect on changing the heart rate in our experiment. So what we're going to do is take a resting heart rate. So you're going to take your research subject, another student within your lab group, hook them up to the pulse transducer, take a recording for 2 minutes, so take their resting heart rate. Then you're going to want to put a comment into the lab chart, have that person run in place or realistically walk up and down the stairs for 2 minutes. And so, don't crash into one another, don't fall, do anything like that. But vigorously get your heart rate up for 2 minutes, come back into the lab, hook up the pulse transducer, and then take the heart rate during a period of recovery. Everybody is going to do that. We are going to collect the observations and put them on the board in the front of the room. We're going to ask you to calculate the mean. And so both in Microsoft Excel and Google, whatever the Google Sheets, the Google version of Excel is going to be, you're going to use the command equals average, and so that's basically a programming command within this range, and this is going to be the range of the data in column B in cells 3 through 12, and that's going to give you the average. You can calculate the range, and so to calculate the range of that same set of values, you use equal max will give you the high, the min is going to give you the low, and so the range is going to be the maximum minus the minimum. And then for calculating the standard deviation, use a slightly different command, equals STDEV, standard deviation, of that same range, where the range B3 to B12, column B, rows 3 through 12, so cell B3 through cell B12 is where we're going to have the data that's going to be entered. And so you're going to calculate those descriptive statistics and then put them in your lab report. We're not necessarily going to graph it in this week's experiment, but you're going to take a look at it, and your results should look kind of something like that. And so heart rate at rest, oftentimes very low. If the person is vigorously exercising, their heart rate is going to increase, and then over time, after 4 minutes of recovery, they're going to get down closer to that rest amount. In this case, the bars are representing the height of our median, our average, and these error bars are showing us plus and minus the standard deviation. So basically, again, getting into a lot of statistics, about 2/3 of the observations, or about 2/3 of the time we do the experiment, we can expect to find results within the area between the top error bar and the bottom error bar. And so we can take a look at this and say, okay, eyeballing it, it looks like, heart rate is going up and coming back down in result of exercising and then recovering. But, eyeballing is only so good. So you want to run some statistics. And so what we're going to do is run a student's t-test. And so you're going to go in, select the function for doing the student's t-test. And if I think about it, I'll post a video that'll go along with this. You select data set 1, select data set 2, pick the appropriate number of tails. And so is it a one-tailed test or a two-tailed test? Two-tailed test is always going to be appropriate. It assumes that the difference could be in either direction. And so the change in the independent variable could be either increasing or decreasing the dependent variable we want. If you have good, solid evidence that it's only going to be on one side, the treatment that you're doing, the change in the independent variable that you're doing is only going to do one thing, it's either going to increase it or not, then it's possible to do a one-tailed test. But you need to have justification to be able to do that. Now, for the experiments that we're doing in this week, we are taking a look at the same subject before and after exercise, or during exercise and at rest. And so we're going to be doing a paired t-test. The other types of t-tests are two samples, and so that would be when we have two different individuals, a group that's treated one way, a group of individuals that are treated another way, and it would be either two samples with equal variance or two samples with unequal variance. And we'll talk about those if the situation arises in lab. And then what we're going to ask you to do is complete the analysis questions. So at the end of each lab, we'll be a series of questions that you need to be able to answer based on your observations from the lab, based on your reading of the lab handouts, to answer those. And again, those are going to be kind of similar to what we're going to see on the lab exam coming up. Once all of that is completed, you're going to want to save your lab report and then upload it. And so underneath the template is going to be a submit lab report here. And so you basically upload your file at that location. If you have a good data set, I'm asking, just because we're trying to collect more and more kind of stock data that we have available. If you've got a good set, you can go in and go File, Save As. This will save it in the ADICHT format. That's the format for the lab chart system, and you can submit it. It's not required. We're starting to collect it, so it's not, there's not a huge need for lots and lots of these, but if you've got a good data set that's well labeled, please consider uploading it here so that we have that available in future years. As always, your laboratory instructor may have further details and updates for you, so please check in with them. And as always, if you have any questions or concerns, please do not hesitate to reach out to me either as a lab coordinator or your laboratory instructor. Thank you, and have a great week in lab this week.